Tehetséges dear students. First, I would like to show our department, the Department of Pharmacology, which is a large department in the Faculty of Medicine, and we are working on different topics, mainly on cardiovascular pharmacology and physiology. And this is the lab that Tamás is working, facing the windows facing on the square, and the other side is just the garden. And this is all painted by a professor of surgery, exactly the same year that I was born. So as you see, not too much changes in all the long second. So in our laboratory, we deal with cardiac cellular electrophysiology, and this is mainly concerns potassium channel physiology and pharmacology. So these are very systematically indicating the potassium currents, which helps to repolarize the cardiac cell. And many millions of cells, the action potential, make up the surface ECG, which cardiologists see in the everyday practice. If something goes wrong, there will be a problem with the heart, which can be lethal, and regularly can develop cardiac arrhythmia, fibrillation, then the heart stops, and it takes several minutes. This is eventually death. So this is a major problem in medicine. So that in pharmacology, it's a very important area to somehow understand the mechanism of arrhythmia and develop anti-arrhythmic drugs, and somehow to understand the mechanism of drugs which produce arrhythmia and sudden cardiac death as a side effect. And the other topic is, which is closely related with this subject is the electromechanical coupling, which is one of the major mechanisms which controls and connects electrophysiological events with the contractile functions. So in our lab we use the conventional microelectrode technique, which measures the potential response on excitable cardiac cells. This is a potential response, and we use a very old-fashioned technique for that, but that's a very useful technique. The microelectrode indicate the cells, and with the reference with the electrode measure the potential difference between the inside the cell and in the extracellular matrix. And this is very simple setup, but it's a very good pharmacological techniques for it. And when we look behind the potential response of the cardiac cell, then we look at the individual ionic currents which flows through membrane protein-based ion channels, and this is done by the patch clamp technique. And the first step to do patch clamping in cardiac myocytes is to isolate the cells. So this is the process of cell isolation, which we do in the lab and under the microscope control patch pipe, and try to measure the currents. And this is the schematic diagram for that. So, and as you can see, we have a microcyte and the cell, and we control the potential of the cell to an amplifier, and then we measure the current which is necessary to maintain that given potential, which we enforce the cell by the amplifier, and everything is nowadays computerized. The setup for that a little bit more sophisticated, and actually is Professor Varró who invented this technique, and got Nobel Prize for this very important technique. It's regularized cardiac, and not only cardiac, in the wide spectrum electrophysiology in general, and we visited Szeged, and we visited our lab, and just talking with the students. So when I would like to introduce our Szentgyörgyi student Tamás Hézső. He was finished the secondary school here in Szeged, in the Radnóti Miklós High School, and he won different sort of competition in chemistry, and also he had participated in Singapore in the International neurological he has interested in cardiology, so he joined to our lab, and he had I gave him a very simple, first of all, a simple task, just to learn the patch clamp technique, which is a sophisticated technique. It takes months for the PhD student to master it. He should do that independently, so that was quite difficult, and because we got an easy project to compare the one type of current between different species. He is very worried here, and not only because he will have the presentation, but because it has a very difficult week, because after this lecture, I think in Wednesday, he will have the final exam in anatomy, which one of the hardest exams in the medical during the medical course. So the current which he will present, some of the data which he himself gathered, is the sodium calcium exchange current, which is through a pumping mechanism, which is pumping pump calcium out from the cells, and it's coupled to free sodium moving units, generate an electrical current, which can be measured by the patch clamp technique. And I just would like to point out that there are no selective inhibitors. There was no selective inhibitor until now. We had now a unique opportunity to have an organic compound for that, so that makes us easy to study this current. And this plays an important arithmetic and which is behind the mechanism of sudden cardiac. That's why I would like to ask Tamás and just give some word about this work which he performed now.
Dear audience, I would like to talk about the possible species differences in cardiac sodium calcium exchanger. This topic is important to understand better the mechanism of arrhythmogenesis. Actually, I started to work in electrophysiological lab in Department of Pharmacology and Pharmacotherapy half year ago. To talk about first aims, actually I decided to get experiences with the whole cell patch clamp technique method. I decided to learn how does it work, and I had to learn how to perform it. Actually, I will introduce the tools and the methods. As a beginning, my mentor advised me to make a comparative analysis of sodium-calcium exchanger in different species like dog, rabbit, and guinea pig. First of all, as I mentioned, I use whole cell patch clamp technique. In this technique, we should use single ventricle myocytes. The first step, we have to isolate the cells. As you see in the first picture, modified Langendorf column is used to perfuse the hearts with oxygenated solution at 37 Celsius degrees. In this time, we use several enzymes like collagenase, protease, elastase, and here you can find the final solution and the single ventricle myocytes. I think this is the hardest part of my job because the cells have really, really bad conditions most of the time. Single ventricle myocytes are obtained by enzymatic dissociation of mammalian hearts. Secondly, I would like to introduce the pipette puller. As you see in this picture, you can find the pipette puller, and it heats and pulls this glass tube, and the tip diameter of micropipette is approximately 1 micrometer. Here you can find a picture about this machine. I would like to introduce also the patch clamp setup. As you see, you can find here the microelectrode. You can find referential electrode, the patch clamp amplifier, and the computer. I had to learn how to use this software, and I can tell you that it's quite hard to make my own protocol or edit a protocol. Actually, we use these programs that it's not really necessary. To tell about the whole cell patch clamp technique, as you see, we use this small micropipette. It's really tenuous. And here you can find the microsite. First of all, we should suck a small piece of membrane onto the tip of a glass micropipette, and after that, between the glass and the cell membrane, you can find a really huge interaction. After that, you will get a gigaohm seal, and finally, you should try to apply more suction to rupture the cell membrane, and you should try to open the cell. After that, the intracellular solution and the cytoplasm will mix. As I mentioned, we measure in this whole cell configuration, and we try to open the cell in this thyroid solution, but after that we will use several special solutions like intracellular solutions and extracellular solutions to block the other currents. We use cesium hydroxide, tetraethyl ammonium chloride, barium chloride, wabain, lidocaine, and nizolipine. And it's also important to make an exact free intracellular calcium level in the cell. To talk about the measurement of N6 current in guinea pig cells, in the first step, as you see, we put a thyroid solution, and after that, we should try to block the other currents with the blocking solution. And finally, we put 10 micromole nickel chloride to block 100% of N6 current. In the D graph, you can find the difference between the B and C graph. Actually, this is the nickel sensitive N6 current. And in this graph, you can find the characteristic of N6 current in different species. As you see, we measured in guinea pig, human, dog, dog, and rabbit cells. To compare and contrast them, as you see, you can find the biggest amplitude in guinea pig cells. To make another comparative analysis, the density of N6 current in different species, we measured in reverse and forward mode the N6 current, and as you see, where you can find normalized data and average data. Normalized data to cell size. As you see, you can find the biggest amplitude also in this line. Also in reverse mode and also in forward mode. Conclusion and implication. Density of N6 current significantly bigger in guinea pig cells comparing to human, rabbit, and dog cells, which may have importance in the understanding of species differences of the electromagnetic coupling and cardiac repolarization. Thank you for your attention.